When you think about it, the story of humanity isn't just written in books or carved in stone. It's actually written in the DNA of the animals all around us. It's this incredible kind of hidden science that connects our most ancient ancestors to the most advanced labs of today. It's a science that is literally rewriting the code of life itself. So let's dive in.
So let me start with a question for you, and it might sound a little bit like a riddle. What on earth is the connection between your loyal pet dog, something from our ancient past, and a virus that's been engineered to kill cancer, something that sounds like it's straight out of a science fiction movie? Well, the answer, it's probably not what you think.
The answer to that riddle, it all comes down to a field called animal biotechnology. And believe it or not, this is a story that doesn't start with test tubes and lab coats. Nope. It starts with the very first humans who tamed a wolf, setting the stage for the dogs we have in our homes today. And that same story stretches all the way into a future where we can program a virus to hunt down and cure diseases like cancer. It's really the story of how we've learned to work with biology itself.
To really get a grip on where we're going, we first have to understand where we've been. And this incredible journey, it started thousands of years ago. Not in some sterile laboratory, but out on the farm.
That's right. The very first biotechnologists, they weren't scientists, they were farmers. By just choosing which animals to breed, the ones that were friendlier, or gave more milk, or had warmer wool, they were basically performing a kind of slow-motion genetic engineering. They were shaping entire species over centuries to better fit our needs. This was really the dawn of us learning to manipulate the code of life.
So for thousands of years, progress was pretty slow, pretty steady. But then, a series of breakthroughs just lit a fire under everything. First, you have Gregor Mendel figuring out the basic rules of heredity. Then, Watson and Crick give us the actual blueprint of life, the DNA double helix. A few decades later, Dolly the sheep comes along and proves we can copy that blueprint. And now, in just the last few years, we've developed tools like CRISPR, which let us not just copy the code, but precisely edit it.
And what's so wild is when you see it visualized like this. Look at that chart. You see that long, basically flat line for thousands of years of discovery? And then, boom. In the modern era, that curve just skyrockets, almost straight up. The pace of discovery isn't just fast, it's become truly exponential.
So, after all that history, where did it lead us? Well, it brought us to our modern definition of animal biotechnology. Today, it's all about taking that ancient idea of shaping life and supercharging it with powerful molecular tools to genetically engineer animals for all sorts of specific, useful reasons.
And this isn't some narrow, niche field. Just look at the UN's definition. We're talking about any application that uses any biological system. It's a fundamental new way of working with life itself to create things we need. The potential here is just almost limitless.
Okay, so that's the big picture. But where is this technology making a really profound impact? Well, maybe nowhere more so than in medicine. We are using these tools to heal our own bodies in ways that, honestly, used to be completely unimaginable.
Some of these breakthroughs sound like they're straight out of science fiction, but they are happening right now. For instance, researchers are taking simple skin cells and turning them into complex, functioning, kidney-like organs in a lab. Just stop and think for a second about what this could mean for ending organ donor waitlists. It's incredible.
And this isn't just theory. It is saving lives today. Take this story. A young boy with a horrible genetic skin disease was given a new chance at life. How? With sheets of what's called transgenic skin grown from his own cells, but after they were genetically corrected in the lab. This is gene and cell therapy in action, right here.
And get this, we're even turning our own bodies into medicine factories. This therapy literally edits a patient's own stem cells to make them resistant to HIV. We are fundamentally changing our own biology to fight diseases from the inside out.
So what this all adds up to is a brand new medical toolkit. We're moving way beyond just treating symptoms. We can create custom vaccines, we can treat genetic disorders at their source, and we're even exploring using genetically modified animal organs for human transplants. That's a field called xenotransplantation. It's a complete revolution in how we even think about healing.
But this revolution, it's not just happening in our hospitals, it's also happening out in our fields and in our barns, and it's fundamentally changing how we're going to feed a growing planet.
So how does this work on the farm? Well, for centuries, we've known there was incredible genetic potential in livestock. Just look at this historical photo of this absolutely massive pig. What biotechnology lets us do now is unlock that same kind of potential, but not by chance. We can do it with precision. It's less about making animals bigger and more about making them genetically healthier and more efficient. And just to put that in perspective, that pig from back in 1933, it weighed over 1,157 kilograms. It just goes to show you the kind of amazing genetic potential that already exists out there in nature. Modern biotech isn't about creating extremes like that, but about understanding the genes for good traits, things like disease resistance or better growth, and then enhancing them in a really targeted and sustainable way. The whole idea here is to engineer a smarter, more efficient food supply. We can help create animals that grow faster, that are naturally resistant to horrible diseases, or that produce more nutritious food, like enhanced milk. It's a total reimagining of what agriculture can be.
All right, so we've seen the hospital, we've seen the farm. Now let's look out to the horizon, where, honestly, the line between science and science fiction is getting blurrier every single day. And at the absolute heart of this future is a technology called CRISPR. The easiest way to think about it is like a find and replace function for DNA. It lets scientists find a very specific, maybe a problematic gene, snip it out, and then replace it with a healthy version. This is the exact technology that makes those cancer-fighting viruses we talked about possible. You can edit a virus's genetic code so it only targets and destroys tumor cells while leaving all the healthy cells alone. It's incredible.
And if you thought that was wild, just take a look at this. What you're seeing here is a lamb, and it's developing not inside its mother, but inside of a biobag, basically an artificial womb. This technology, it's called ectogenesis, and it challenges our most fundamental ideas about where life comes from and how it can be sustained.
Which brings us right back to this graph, one last time. We are living on that almost vertical part of the curve, a period of explosive, totally unprecedented change. The pace is just mind-boggling. And that little dotted line stretching into the future, that poses the biggest question of all. Where is this all going to lead?
So in the end, we've gone from simply watching life to guiding it, and now to actually rewriting its most basic instructions. The power is, quite literally, in our hands. The only real question left is, what kind of story, what kind of world are we going to choose to write with it?